Hello guys, I'm starting with plant physiology. You already know that the sequence of events happen in the plants. The first step is LHC complexes absorb the photons. Next, light reactions happen. Next, Kelvin cycle. Next, glycolysis. Next, Krebs cycle. Next, oxidative phosphorylation. So I am starting with LHC complexes, which is first one. All of living organisms getting the energy from the solar light. Energy comes as photons. These photons are absorbed by the light harvesting complexes. There are several molecules. They are chlorophyll-a, chlorophyll-b, carotenoids. These molecules absorb different wavelength photons. I am going to read out for you. Introduction to light harvesting complexes. Light harvesting complexes are pigment protein complexes present in thylakoid membranes of the chloroplast that absorb light energy and transfer it to reaction centers of the photosystems. So LHC molecules present in thylakoid membranes, okay, and absorb the light energy and transfer them to photosystems. Main role: capture photons efficiently, expand absorption spectrum, transfer excitation energy to reaction center chlorophyll, P680, P700. So electrons are transferred to chlorophyll here. There is two reaction center, that is P680 and P700. The second one is types of light harvesting complexes, LHC2, associated with photosystem 2. Most abundant membrane protein on the earth contains chlorophyll-a, chlorophyll-b, carotenoids. These are all names. I cannot explain you everything. You have to mug up. The third point is forms trimers. Located mainly in grana stalks. Grana part is located inside. Functions: collect light energy, protect against excessive light. So it takes energy, also protect it from excessive light. Next, LHC1 associated with photosystem 1. Smaller antenna size, located in stroma lamellae. Transfer energy to P700. In this slide we can see where is stroma, where is grana. In this slide we can observe that stroma is near to the outer membrane and grana is inside the chloroplast. We can see chlorophyll-a structure and we can see chlorophyll-b structure. Later we will observe what things are different. First let us see chlorophyll-a structure. Let us see main structural parts. You can see that there is magnesium ion in the center of chlorophyll-a. There is 4 pyrrole rings. You can see in the diagram. Also, this magnesium ion is responsible for absorbing the light. Also, it stabilizes the electron. Okay, you can study this. The upper part of chlorophyll-a is large ring made up of 4 pyrrole rings. You can see in the diagram. At the center, there is a magnesium ion. You can see magnesium ion. This magnesium ion is important for stabilization of electron. Also for allows light absorption. This ring contains alternating double bonds. You can see double bonds here, double bonds. Okay, na? One single bond, next double bond, one single bond. You can see as that. The ring contains alternating double bonds, creates conjugated system, which is responsible for absorbing red and blue light. These are responsible for absorbing red light and blue light. There is a tip for CSIR-NET. The porphyrin ring is often called a chlorine ring in chlorophyll, because one double bond is reduced compared to porphyrin. So here double bond is reduced so that it is called as chlorine ring. Next let us see phytol tail, membrane anchor, okay, a long hydrophobic hydrocarbon chain. We can see in the next slide. You can see the phytol tail. Here it is long chain. Let us see side groups. Chlorophyll-a contains methane group at key position on the ring 2. This small chemical detail differentiate into chlorophyll-b. So this methane group differentiate the chlorophyll-a and chlorophyll-b. You can see the methyl group here. There is no methyl group here. Functional meaning: because of this structure, chlorophyll-a absorbs mainly 430 nanometer, which means chlorophyll-a absorbs mainly 430 nanometer, which is blue, and 662 nanometer, which is red wavelength. It acts as the reaction center pigment P680 in PS2 and P700 in PS1. You should carefully observe the chlorophyll-a and chlorophyll-b structures. Let us go to next slide. Key structural differences from chlorophyll-a. The overall structure is almost identical. We already know that chlorophyll-a contains methyl group and chlorophyll-b contains aldehyde group. Same tetrapyrrole ring. Okay. Same central magnesium ion. same phytol tail. But one crucial change exists in chlorophyll-b. The methyl group is replaced by aldehyde group. This tiny chemical change causes slight shift in electron distribution. Different electron absorbs properties. Effect on light absorption. Because of aldehyde group, chlorophyll-b absorb more green light, which is 453 nanometers. It slightly varies, but remember 450. This expands the range of usable sunlight for photosynthesis. So this is useful for photosynthesis. Let us go to next slide. I again mention easily, so please watch it later. So let us see second one, carotenoids in light harvesting complexes. What are carotenoids? Definition. Carotenoids are accessory pigments present in light harvesting complexes that assist chlorophyll in capturing light and protecting the photosynthetic apparatus. So carotenoids are useful for absorbing light, also for protecting the photosynthetic apparatus. They absorb mainly blue and blue-green wavelengths, that is 400 to 500 wavelength nanometers, where chlorophyll absorbs less. Types of carotenoids and carotenes. Hydrocarbon carotenoids. The second point is types of carotenoids. Carotenoids are two types, carotenes and xanthophylls. These are mainly hydrocarbon carotenoids, example beta carotene. They are in orange to reddish color. They are found near the reaction centers. The next one of the carotenoids is xanthophylls. These are oxygen containing carotenoids, examples lutein, violaxanthin, zeaxanthin. These are yellow colored pigments. These are important for photoprotection. Third point is structure of carotenoids. Carotenoids include long pollen chain with conjugated double bonds. Carotenoids contains hydrophobic molecules embedded in thylakoid membrane. This conjugated system allows absorption of high energy light. So carotenoids are useful for high energy light. Here I mentioned that conjugated double bonds are useful for absorbing light energy. The fourth point is functions of carotenoids in LHC. LHC harvesting role. Captures blue light energy, transfer exciton energy to chlorophyll-a, expand the absorption spectrum. So it absorbs the blue light, also it transfers that energy, which means electrons, to the chlorophyll-a. The next point is photoprotection role. We already mentioned so many times that it protects the photosystem. So photoprotection role. Carotenoids prevent the damage of excessive light. They perform triplet chlorophyll quenching, prevent formation of singlet oxygen, that is ROS, protect photosystem proteins. If there is no carotenoids, then photooxidative damage will be happen to the chlorophyll. The next point is role in non-photochemical quenching. When light intensity is very high, carotenoids convert excitation energy to heat. Okay, prevent over excitation of photosystem. Key pigment zeaxanthin. So zeaxanthin do these actions. We already discussed that carotenoids include carotenes and xanthophylls. You can see the structure. There is alpha carotene, beta carotene, gamma carotene, epsilon carotene, lycopene. There are different types of xanthophylls: fucoxanthin, cryptoxanthin, canthaxanthin, astaxanthin. In previous slide we have discussed that triplet state protects from the sunlight. If there is excessive light, chlorophyll enters into triplet state. This triplet chlorophyll produces ROS, that is singlet oxygen. Next we have also discussed about quenching. Quenching means processing of excessive energy to heat. So now we already know that carotenoids prevent photooxidative damage. We can see that in the diagram we can see that chlorophyll here is the photon energy. Next to chlorophyll is excited. Triplet chlorophyll, which means 3 chlorophyll ions are formed. Next this energy is converting to heat. Next here you can see that oxygen is getting excited energy and ROS process is happening here. Here you can see that carotenoids also getting the excited energy and this excited energy also getting out by heat. In this slide we can see that violaxanthin structure, anthraxanthin structure, zeaxanthin structure. Zeaxanthin helpful for heat dissipation pigment. You can see one thing. Here pH value is 5.8. Here pH value is 7.5. So let us come to the point. If there is highlight, lumen pH decreases. We can see here highlight and low pH. If there is low light, high pH. Here we can see the light energy comes from viola-xanthin to anthoxanthin and zeaxanthin. So here energy flowing from viola-xanthin to zeaxanthin. Zeaxanthin helps in photochemical quenching. We have already discussed about quenching. So zeaxanthin helpful in quenching. The next topic is energy transfer and light harvesting complexes. In intermediate level, we have studied about PS1 and PS2. First, photons hit the chlorophyll-a or chlorophyll-b. Okay. Here, let us see chlorophyll-b excited. Excited energy migrate to chlorophyll-a. Again, excited energy going to reaction center. We can see that excited energy from the accessory pigments, that is chlorophyll-b or carotenoids. So, this energy moving to reaction center. Reaction center may be P6CIT or P700. Let us watch about this all in next classes, that is PS2, PS1, how Z cycle happens. Let us see later. Non-photochemical quenching in light harvesting complexes. We already know that what is quenching. What is non-photochemical quenching? We already discussed about this. Non-protective quenching is protective mechanism where excessive absorbed light is safely released as a heat to prevent the damage of photosynthetic apparatus. We have discussed it. So let us move further. Role of xanthophyll cycle in NPQ. We have already discussed about this topic, but this is in little depth. Let us see types of NPQ. Simple net version. QE, energy dependent quenching. QT, state transmission related. QI, photoinhibitory quenching. Key proteins involved. PSB S protein. Senses pH changes in thylakoid lumen helps in activate NPQ process. Please remember this all. I cannot explain you everything. These are not explainable. We can see clearly here. Please watch it in big picture. There is excessive light. Viola-xanthin absorbs it and next it moves to zeaxanthin. Here energy converted to heat. State transitions in light harvesting complexes. If there is excessive light in PS2 or PS1, they get balanced in both by using state transitions. So let us see the definition. State transitions are regulatory processes where LHC2 antenna complexes move between PS2 and PS1 to balance the excitation energy. Purpose: maintain equal energy distribution between two photosystems. I already told you that. Why state transitions are needed? Sometimes PS2 absorbs more light than PS1. PS1 absorbs more light than PS2. So here imbalance is happened. So in order to maintain this imbalance, we need this state transitions. You can see in the diagram. State 1 normal condition. State 2. State 2 LHC moves towards PS1. Because PS2 absorbed high energy. Because PS2 absorbed more energy. So these transitions are happening here. Let us read the points. Mechanism of state transitions. How this happens. State 1 normal condition. We can see the normal condition. LHC2 mainly attached to PS2. Balanced excitation energy. We can see LHC2 here. It is attached to PS2. State 2 PS2 overexcited. So high energy excited the PS2. Plastoquinone pool becomes reduced. We cannot see all of them here. We can see STN7 kinase is activated here. LHC2 ions get phosphorylated. We can see phosphorylation happening here. Some LHC2 moves towards PS1. Here it is written. PS2 moving towards PS1. Reverse process. When PS1 become excited. If here energy get more, this energy moves towards PS2. So balance get happen in both. So remember this STN7 kinase is key enzyme here. It controls phosphorylation of LHC2, central regulator of state transitions. So it also performs the transitions as well. Organization of LHC2 supercomplex. What is LHC2 supercomplex? To form the large antenna to PS2 reaction center, these complexes are required. Let us see the definition. LHC2 supercomplex is structural arrangement where multiple light harvesting proteins surround the PS2 reaction center to form large antenna system. Main function: to maximize the light capture and transfer energy to PS2. Please remember this all. I cannot explain you everything. So to receive more light, these super complexes are required. I read out for you. Structural components of LHC2. Major antenna proteins. LHCb1, LHCb2, LHCb3. Form trimeric LHC. These are helpful in forming trimeric LHC2 complexes. Contains chlorophyll-a, chlorophyll-b and carotenoids. So these are major antenna proteins. Okay. There is also minor antenna proteins. Let us see them all. CP29, CP26, CP24. These are located close to PS2 core. Help regulated energy transfer. Important in photoprotection. So remember this all. This is the last slide. Arrangement of around PS2. PS2 core is present in the center. Minor antenna proteins surround the core. Major LHC2 trimers are attached to outside. This arrangement creates large light harvesting surface. We have discussed in previous slide there is major and minor proteins for super complex. So they are telling that PS2 is in the center. We can see here. There is major and minor complexes around here. These are helpful for absorbing maximum light. Functional importance increases the absorption of cross section, controls antenna size, helps in state transitions, participants in NPQ. Please remember this all. This is first lecture to me. I might not have taught you well. I improve myself in further lectures. Please follow my channel for more videos. Thank you for watching.